Hi everyone, welcome to your chapter 15 lecture video. If you are watching this in the fall of 2020, quick reminder that October 23rd, which is a Friday, you do have homework 4 due. Okay, it's already posted. It's in the week 10 folder. So just make sure that you get that done by Friday. It will cover chapters 15 and 16, okay? Okay, so let's think about where we've been and where we're going in chapter 15. So our last couple of chapters focused on the cell cycle, cell cycle, mitosis, and meiosis, right? So in thinking about what was going on, essentially what we were doing is making copies of our genetic material and then distributing them among daughter cells. If we're talking about mitosis, then we were distributing these copies among somatic cells. If we're talking about meiosis, then we were undergoing 2 rounds of cell division to make gametes. But in the end, we were still distributing copies of DNA among daughter cells. So what this chapter really focuses in on is this process of how we begin to copy DNA in the first place, how we make it, and then should any mistakes be made during the copying, how we repair those mistakes. Okay, and there are 3 things that I really want us to focus on in this chapter. The first thing that I want us to think about is how do we know that DNA is actually the hereditary material? I mean, we've all been hereditary. It's hard to talk. Hereditary. But hereditary stuff. We've all been taught for so long that DNA is what genes are made of. You've probably never stopped to question how it is we know that genes are made of DNA. So we're going to talk about that first and foremost. Then the next thing that we're going to do is we're really going to hone in on how DNA replicates. Okay, how does it get copied? And what we're going to do is we're going to talk about the series of events. You're going to need to know the names of some enzymes. So you're really going to need to study the process here, okay? And it's not terribly difficult. And then the last thing that we will focus in on is the repair. Once it's replicated, if there are any mistakes, how can repairs be made? Okay, so let's first think about this question here. How do we know that DNA is the stuff of heredity? Now let's take a step back. We talked about our buddy Gregor Mendel in our last chapter. So remember that Gregor Mendel, and I'm just going to put Greg, our buddy Greg, he was working in the late 1800s, and about 1865 is when he published his paper documenting the rules of heredity. And if you remember from our previous lectures, Greg did not know much about genes at the molecular level. Everything that he deduced based on observations of physical traits. And then, if you remember, his work was promptly forgotten until about 1902 or 1903, when we had 2 scientists who independently were able to use a microscope, visualize the process of meiosis, and in looking at meiosis, they recognized that what was happening to the chromosomes really matched up with the predictions made by this obscure monk in 1865. And so these 2 guys, if you remember, they are credited, it's Sutton and Boveri, and they're credited with developing the chromosome theory of inheritance. So the problem was this. If you think about chromosomes, then you have to remember that chromosomes are made up primarily of 2 things. So I'm going to draw this little diagram. I think you guys will remember what I'm talking about. I'm going to draw this here. And thinking about how the DNA is arranged in your nuclei, it doesn't occur as DNA alone. It occurs as DNA. And if you think about DNA like a thread, then it's a very, very long thread that would be vulnerable to knots. So what we do in our nuclei is we wind this thread around some proteins. And these proteins, if you remember from previous lectures, these proteins are called histones. This combination of DNA plus proteins is called chromatin. And this is the stuff that chromosomes are made of, right? So what happens during meiosis or mitosis in prophase is that these histones begin to stack on one another to condense down into those stereotypical X-shaped chromosomes that you're used to seeing. But the thing that I really want you to focus in on is that chromatin is made of both DNA and proteins. So what early scientists knew is that chromosomes were likely the thing that were passing on traits. But was it the DNA in the chromosomes? Or was it the proteins in the chromosomes that might be responsible for passing on hereditary information? Now, for a long time, it was thought that the proteins in the chromosomes were likely the culprit. Now, why did we think, I say we, I wasn't around then, I'm old, but I'm not that old, right? Why did scientists of the time think that protein was the stuff of heredity? Well, to get at that, I want you to think about the basic molecular structure of proteins versus nucleic acids. Remember that proteins are made up of amino acids, while nucleic acids like RNA and DNA, they're made up of nucleotides, and I'm just going to abbreviate that with nuke, okay? Amino acids and nucleotides. Now, if you're talking about the different types of nucleotides that can make up a molecule of DNA, you basically have 4 different types, right? You can have a nucleotide with an adenine attached, one with a thymine, one with a cytosine, or one with a guanine, right? You only really have 4 different variations on a nucleotide that you can include in a molecule of DNA. Early scientists looked around at biodiversity. They looked at sheep and dogs and plants and people and said, it just doesn't make any sense that a molecule that's so chemically simple could account for all the variation that we see in traits across all of living beings on the planet. On the other hand, if you think about proteins and how many different amino acids there are 20 amino acids that can be combined to form a protein molecule. And so early scientists look at this information and they said to themselves, this could generate a lot of different types of molecules. There's probably more variety in proteins than there is in DNA. So it makes more intuitive sense to us that protein would be the stuff of heredity. So for a long time, there was this debate about whether it was DNA or whether it was protein that's responsible for passing on genes. And one experiment that really sealed the deal on which was called the Hershey-Chase experiment. This experiment was performed in 1952. Now, there had been some other earlier experiments a few years earlier that sort of hinted that DNA might be the stuff of heredity. But in 1952, these folks really performed an experiment that sealed the deal. And what I'm going to do is get rid of this box here because I want to do some writing and drawing. Okay. There we go. Okay, so let's stop it. Come on, PowerPoint. Nope. Why won't it just, there we go. Okay, so in order to understand the experiment that these folks performed, you really need to understand something about viruses, okay? And the virus that we're going to focus in on for this experiment is one that infects bacteria. It's called a T4 bacteriophage. Now, what's happening with this bacteriophage? A phage is just a virus that infects bacteria the same way that we have viruses that infect our cells. So here's how this works. And so I have a diagram of it for you here, but let's just draw it for the sake of walking through this process. So I'm going to draw a very simplified version of this bacteriophage. And it's got some key characteristics. It's got kind of this top part here. See that right there? It's got a neck that looks a little bit like an accordion. If you zoom in on that, you can see that this neck looks kind of like an accordion. And then it has these little legs. And what happens is this virus that looks kind of like a spaceship, and yes, it really does look like that. What it will do is this outer part here that I've drawn for you, it is called the capsid, and you can think of it as a capsule, okay? Think of it as an empty space, and it is made entirely of protein. Okay, and then on the inside, stored inside this protein capsule, is the DNA, okay? This DNA is viral DNA and contains the instructions on how to make more virus. Now, what will happen is that this virus will use its little legs to attach to the outside of a bacteria like an E. coli. And remember that E. coli is a bacteria that has a single circular chromosome, as bacteria do. They have a single circular chromosome. So what this virus will do upon attachment is it'll basically push down on this and use its little neck like a hypodermic needle to inject its DNA into the E. coli. So then what happens is the cellular machinery of E. coli that gets busy copying its own DNA will also copy the viral DNA and make a whole bunch of new viruses inside the cells. Here you have some new virus being made, and there's the little DNA. And you're going to make a whole bunch of these copies. Eventually, you're going to make so many of these viruses using the viral DNA that's been injected into this bacterium. You're going to make so much of it that this thing will become bloated, and eventually it will rupture and a whole bunch of new viruses will spill out. So that's what's happening here. Let me just enlarge this for just a second for you folks so that you can really see what's going on. Here we have, gonna get rid of this really quickly. Here we have our bacteriophage, okay? This is really what it looks like. It's injecting its viral DNA into the bacteria, and then the bacteria, it can't really differentiate between its own DNA and the virus DNA. So it starts to copy this virus DNA and use that as an instruction to make more viruses. Eventually, so many of these viruses get manufactured that this will essentially cause the cell to rupture. These guys will go out into the environment where they can then infect new bacteria. And just so that you can see that these things really do look like little spaceships, here's what they look like under a microscope. And there they are injecting DNA into the E. coli, all right? So the things that I want you to really take away from this is that there's two parts to this this virus. There's a DNA part and there's a protein part. And that is the key to understanding this experiment. So what these folks did, Hershey and Chase are two different scientists, okay? What these folks did is they say, what we want to look for is whether DNA is getting into these bacteria, because they didn't know, or whether protein is getting inside these bacteria. Whichever one is getting inside these bacteria is causing the bacteria to make more and more copies of the virus, and therefore must be the genetic material. So here's what they did. And I'm actually going to insert a new slide, and I'm going to make it a blank one so that I can draw for you. Okay, so here's what they did. They basically took some E. coli. This is an E. coli cell. They allowed it to become infected with our bacteriophage. And then once this bacteriophage was infected, they took these infected once a bacteria was infected, they took these infected bacteria and they grew them up in two different beakers. One beaker, they placed infected bacteria into. So here's our infected bacteria. And this contained nutrients, including radioactive phosphorus. I'm going to put radio phosphorus. Now, when you see phosphorus, I want you to think phosphorus, phosphate groups, okay? Phosphorus is a major component of DNA. Remember, DNA has phosphates in its backbone. The second beaker, they put some more infected bacteria into. Okay, so here's our infected bacteria. And in this nutrient medium, they included radioactive sulfur. Sulfur is present in proteins, but not in DNA. So we have infected bacteria here, including a medium with radioactive phosphorus. Phosphorus is present in DNA, but not really in proteins. On the other hand, we have a second treatment where we put our infected bacteria in a solution with radioactive sulfur. Sulfur is present in proteins, but not in DNA. So what they then did was then they let this go on and they examined the resulting E. coli to look for where the radioactivity might be. And what they found was that there was radioactivity, I'm going to put RAID here, radioactivity inside E. coli cells from this treatment, but there was not any radioactivity inside cells from this treatment. So what does that mean? That means that the stuff that was getting inside the bacteria, the stuff that was infecting the bacteria, was DNA. Here, there was no radioactivity that made its way inside. So proteins were not infecting these bacteria. In other words, DNA must be the genetic material because it was giving these E. coli the instructions on how to make more virus. Okay? Now, this experiment is really important in terms of understanding that DNA was in fact the hereditary material. It's also an excellent example of scientific reasoning. So if you're having any trouble understanding this experiment, there is a homework problem that's associated with this experiment. There is a little video that's embedded in that homework problem to help you understand it. And I believe that video is also available in the study area of your text, okay? Or it might be embedded in the chapter, but you definitely have a little video that explains this process with the textbook, okay? So let's just recap this experiment here. This is the figure from your text. Viral DNA is radioactive, viral protein is radioactive, depending on which treatment you're talking about. And then they looked to see where the radioactivity ended up. And what they found was that the DNA was radioactive. And it's what was inside cells. And make sure that you can walk yourself through this process, okay? Quick note, I think I said T4 bacteriophage before. It is the T2 bacteriophage. Not really a question that I care that much about, not something you're going to find on an exam, but just to correct my misspeak, bacteriophage. All right. One quick note, I told you on the earlier slide that what they found was that the DNA was inside these cells. The way that they determined that was through a process of centrifugation, okay? If you don't understand that, don't worry about that. We'll talk about centrifugation more in a little while, but just know that the radioactivity was inside the bacterial cells. All right, so we now know that DNA is the stuff of heredity, that DNA is the genetic material. So that was in 1952. At that point, there's a race. And this race was between very famous labs, and they were trying to figure out, okay, we know DNA is the genetic material, but what we don't know is what exactly is the structure of DNA. So they knew what molecules made up DNA, but they didn't really understand how those molecules were arranged. All right, and so there were some famous folks involved in looking at this problem. And in 1953, there were two fellas that published a paper discussing the structure of DNA. Their names are James Watson and Francis Crick. You've probably heard of Watson and Crick, okay? What did they discover? Well, they discovered that DNA is a double helix. Folks did not know that prior to 1953. However, for years we have been giving these two individuals all kinds of credit for discovering the double helix of DNA. And in 1962, they actually won a Nobel Prize for this work. But I'd like to point out that there were a couple of figures that came before them that were really critical in helping them understand the structure of DNA. So one person was doing some work in 1950, and he was a guy named Erwin Chargaff. And here's what he figured out. He was doing some chemical analysis of DNA. Again, he didn't really know the structure, but he knew what molecules made up DNA. And in his chemical analysis, he found a very curious phenomenon. He found that the concentration of adenine in a molecule of DNA was always equal to the concentration of thymine. And he also found that the concentration of cytosine was always equal to the concentration of guanine. So what this did was it allowed Watson and Crick to develop what we now know as complementary base pair rules, okay? So I'm gonna put base pairing, okay? Then in 1951 or 1952, Watson and Crick were doing their work in London at a place called King's College. And a colleague started working there named Rosalind Franklin in 1951, 1952. And what Rosalind Franklin's work centered on was also a chemical analysis of DNA, but she was actually also trying to work out its structure, and she was using a technique called X-ray crystallography. Basically, what she would do is she would dry down DNA until it formed a crystal, and then she would shine an X-ray beam through it, and then allow those X-rays to, as they moved through that DNA crystal, to become diffracted. And when they became diffracted, they would create an image that was a particular shape. And what Rosalind found using a famous photo called Photo 51, which you guys should totally look up, she found that the shape that resulted from her analysis definitely suggested that DNA was a helix. She didn't know if it was a double helix or a tetrahelix, but she definitely had the idea that DNA was a helix. Now, this work was really, really important in helping give a clue to these folks here about the structure of DNA. And this work has been the subject of a great deal of controversy because it's not entirely clear whether or not Rosalind Franklin's data was used with her permission or not. She had a graduate student at the time. What we do know is that her work, her Photo 51, was shown to Watson and Crick, and that they immediately appreciated that what they were looking at was a helix. Now, whether or not that photo was shown with her permission is the question. Her graduate student at the time, who was also male, suggested that she knew about it. Other folks suggested that she didn't. What we do know is that the relationship between Rosalind Franklin and James Watson was extremely contentious. James Watson is very famous for being very racist in his views and very sexist in his views. He actually wrote a book about the discovery of DNA called The Double Helix, where his descriptions of Rosalind Franklin were so egregious that he was asked to edit his book, and his buddy Francis Crick spoke out against the book. So there was a great deal of sexism, of misogyny surrounding the relationship between Rosalind Franklin and James Watson, and also perhaps between her and Francis Crick as well. What we do know is that despite the fact that her work was so important as a clue, she was not invited to contribute in authorship to that paper, and she was not included in the 1962 Nobel Prize. So this has been the subject of a great deal of controversy surrounding the discovery of DNA. So James Watson, Francis Crick in 1953, we finally know a little bit about the structure of DNA. Let's review that structure, because understanding that structure is really going to be key to understanding how DNA is replicated, all right? So let's look at the structure here. Let's first start out with the basic structure of a nucleotide, which is what we have here, okay? Hopefully y'all remember that a nucleotide can be broken down into 3 parts. The sugar, which is what we call a pentose or 5-carbon sugar. So I'm going to go ahead and draw the sugar like this. Now, what we understand when you're drawing a molecule like this is that in each corner, we assume that you understand that there's a carbon. So there's a carbon here, one here, one here, one here, and then also one there. So that's 1, 2, 3, 4, 5 carbons. It's also convention that we number these carbons. We number them starting in a clockwise fashion from the oxygen. So this is what we call the 1' carbon, the 2, the 3, the 4, and the 5' carbon. So that's our sugar. Another thing to note that's really important is that at the 3' carbon of our sugar, we have an OH group. This OH group is going to be very, very important. So remember that if we're talking about DNA, this sugar has a name, and it's called deoxyribose. All right. The next thing to remember is that another part of a typical nucleotide is a phosphate group, okay? And I'm just going to make that a letter P with a circle. But phosphates groups also have some oxygens and hydrogens surrounding this central phosphorus atom, okay? And then the third component of a typical nucleotide is a base, and I'm just going to draw a circle with the letter B. This base is always attached to the 1 carbon, okay? And this base could be any one of 4 options: adenine, thymine, cytosine, or guanine. So just to recap, we have a base at the number 1 carbon. We have an OH group, that's always going to be at the number 3 carbon. And we have a phosphate group that is always at the number 5 carbon. Now, what if we wanted to link these together? What would we do? Well, let's draw ourselves a couple of these. I'm just going to make them kind of small. So here's our oxygen, boop, boop, boop. Let me make that a little bigger. Okay, so here's our phosphate group. Here's going to be a sugar molecule, deoxyribose. I'm going to put the OH group here. Here's another carbon and attached there. Now let's draw another one. An OH group. Boop. Oh, I forgot a base here. I'm just going to put B, and then a C and a phosphate group. Okay, if we wanted to start linking these together, what would happen is that we would take this OH group along with an atom of hydrogen from here, and we would lose some water, in what's called a condensation or dehydration synthesis reaction, and that would link this nucleotide here to this one here, okay? And that's exactly what has happened in this molecule here. What we've done is we have taken our phosphate, let's do this one. We've taken a phosphate here and we've attached it to where there would have been an OH group, right? And we lost some water. So the end result is going to be a phosphate sugar, phosphate sugar, phosphate sugar backbone, okay? So we have a sugar phosphate backbone of DNA here. Now I want you to notice something. In order to add another nucleotide to this strand, we have to have an open OH group, because what would happen is if you wanted to add another one, you'd take a phosphate group that was attached. And let me draw this guy here. Boop, boop, boop. Okay, so this is where the attachment grows, right here. An OH attached to a phosphate. So this molecule can only grow in this direction. I always tell students to think about this OH group as a parking spot. If you have an open OH group, what you have is a parking spot for the next nucleotide, okay? So we can grow in this direction. And if you think about this direction, you can name it by the carbons. So this end up here, where you have a phosphate group attached, it's actually what we call the 5' end because it's closest to the 5' carbon on the sugar. And then down here, we call this the 3' end because this OH group is closest to this 3' carbon on the sugar. So an important thing to note on the structure of DNA is that it can grow only in the 5' to 3' direction. That's the only way a DNA molecule can grow. Okay, now let's take a look here at double-stranded DNA, because if I go back here, what I've shown you is one strand, right? And we know that DNA occurs in 2 strands and that those 2 strands form a double helix. Now, let's just look at the 2 strands by themselves. So we have our 5' end up here, and we have our 3' end down here. This red line right here is meant to represent the phosphate sugar backbone, okay? And then we have our bases that jut off of our phosphate sugar backbone like that. Now let's look at how the opposite strand is oriented. This one is oriented 5' to 3', but its sister strand is upside down relative to the original strand, 5' to 3'. And when you get into genetics, what you'll find is that the reason for this is so that these bases can pair up. They only pair up upside down to each other. So we say that DNA has something called an anti-parallel structure. Okay. Once we have these 2 strands oriented anti-parallel to each other, then it is just part of their chemistry that they will automatically twist to form this double helix. Now, one quick thing that I want to note is that the sugar phosphate backbone, that is a permanent chemical linkage. That is a covalent bond between the sugars and the phosphates, okay? It's a covalent chemical bond, and it's actually called a phosphodiester bond. I'm gonna split that up. So this part here that we have in the red as we move down, those sugars and phosphates, those things are stuck together really nice and tight. On the other hand, the force that's holding together each of these base pairs is not a covalent bond. Instead, it is a hydrogen bond. Hopefully you guys remember that from our early chapters. And if you remember, hydrogen bonds were not permanent. They resulted from positive and negative charges on the molecules. And so you could think of them as almost like magnetic bonds. And just like a magnet, these things can be pulled apart and stuck together, and pulled apart and stuck together. So that actually is going to be a big part of how we copy this DNA molecule. What we're going to do to copy this DNA molecule is we can actually unzip the DNA strands here and pull them apart. The sugar phosphate backbone would stay intact, so you'd have something like this. And then here, that would stay intact, but you could pull apart the base pairs just like you could pull apart two magnets. And so that really suggests a mechanism for how you could copy this. If you can pull this guy apart into two separate strands, then it's pretty easy to imagine that each of these strands can serve as a template to build a new strand of DNA, right? And so that is exactly what we're going to find in part 2 of the lecture. Talk to you soon. Bye-bye.